require energy input. And the source of our energy input is ATP. Okay? So that's anabolic. Oh, smaller molecules to bigger molecules. For example, your glucose is put together to form glycogen. And then fatty acids are put together to form a neutral fat or triacylglycerol. And your protein is made up of amino acids put together. So that's anabolic pathway. Now, the catabolic pathway is the opposite of anabolic. So catabolic is the breakdown of bigger molecules to smaller ones. And in so doing, we always release energy. If you remember, and we call this your pathway that gives off energy is exothermic. Okay? Exothermic. So we have several of these pathways, the breakdown of our metabolic molecules, or what we call fuel molecules. Now, in case of carbohydrate, the breakdown of glucose down to carbon dioxide, water, and of course, ATP that is formed. Okay? And fats are broken down into fatty acids and glycerol. And the fatty acids are further degraded in the beta oxidation in your mitochondrion. And acetyl-CoA are formed. Acetyl-CoA goes into your citric acid cycle. And the citric acid cycle produces ATP. And we call that substrate level phosphorylation. And the reducing equivalence NADH goes into the respiratory chain, and the respiratory chain oxidizes your NADH to form ATP and carbon and water, not that carbon, and water. No. Okay. Likewise, also proteins are broken down into amino acids, and amino acids can be utilized as fuel. Now, in the you still remember the lecture of Dr. Ryan that the heart uses fuel like ketones, fatty acids, and glucose. No? The heart is very versatile because it can use any kind of fuel. Unlike your RBC, which can only use glucose as fuel. It cannot use fatty acids. It cannot use ketone bodies to furnish energy for the maintenance of the RBC. Okay. So we will be talking about that when we take up the pathways. Like I mentioned already, glycolytic pathways, citric acid cycle, pentose phosphate pathway. You have your beta oxidation of fatty acids for the breakdown of fuel molecules to form energy. Then the other pathway, the third one, is amphibolic. Amphibolic pathway occur at the crossroads of metabolism, acting as a link between anabolic and catabolic pathways. And a good example is your citric acid cycle. The intermediates in the citric acid cycle can be broken down to form or to produce energy. And if you remove all of these intermediates in the citric acid cycle, then there won't be any more intermediates. So the citric acid cycle will not work. This is replaced. Intermediates are replaced. And we call this amphibolic materials, which replaces intermediates in the citric acid cycle so that the citric acid cycle can continue its work. Okay, example. One of the intermediate in your Krebs cycle is alpha ketoglutarate. Alpha ketoglutarate is the carbon skeleton of glutamic acid. So when we say carbon skeleton, meaning to say we remove the amino group of the amino acid. So in this case, glutamic acid minus the amino group, it becomes just an organic acid, ordinary acid, no? And that is alpha ketoglutarate. And that can be utilized in your Krebs cycle. And alpha ketoglutarate also can also be glutamic acid again if an amino group is added to it. So it becomes glutamic acid. Yeah? So that's what we call amphibolic materials, like oxaloacetate, which comes from aspartate, no? And your pyruvate from alanine. So those are amphibolic materials. And the pathway there is known as amphibolic pathways. Okay? Right. Let's continue. It says here there is a constant requirement for metabolic fuel throughout the day. So average physical activity would require 40 to 50% over the basal or resting metabolic rate, or your BMR. However, most people consume their daily intake of metabolic fuels in 2 or 3 meals. 2 or 3 meals. So there is a need to form reserves. In case of carbohydrate, it's glycogen stored in the liver and in the muscle. And then you have lipids, and that is triacylglycerol, that is in the adipose tissue. And protein. Protein can be as part of your structure, especially the muscles. Now, the preference of the cells, they always use glucose first. Then when glucose is low or is deficient already, then fatty acids are utilized by the cells. And after that, it's the protein. So muscles are utilized. In that case, you become so thin or emaciated, and there's a large amount of wasted proteins, and eventually death follows. Unless, of course, there will be a supply of those metabolic fuels. Okay. Now, if there is plentiful, then this is stored. When there's plenty of glucose, it is stored as glycogen. the liver, and your muscles. If there is still extra, then the glucose is turned into fatty acids. This is a very good, this is an area where it has to be understood, especially those who are already obese, that it is not fat that makes you fat. It is carbohydrate, especially your sugar. Anything that is sweet, no? That's your sugar. Uh, there's a book on about sugar. The title is, I could not remember the title anymore. It is White, Sweet, and Deadly. White, sweet, and deadly. Why deadly? Because if you indulge so much on sugar, especially white sugar, it brings about obesity. It is not the fat that makes you fat. It is the sugar. Now, especially now that almost everything in our diet, especially fast foods, there is always sugar. Those who cannot eat without soft drinks, you better cut it down. No? Even our food now are invaded by sugar. They put sugar in order that it will taste better. And you become addicted to it. Especially this, what you call soft drinks. I always tell people that there's nothing soft about soft drinks. If you only know the composition, your cola, the composition there is phosphoric acid. And you know phosphoric acid is a strong acid. It's a very strong acid. In the Western world or in the United States, if you are familiar with this police series, Chicago Police, New York Police, no, in the TV, they carry with them in their cars, their patrol cars, liters of the soft drinks, the Coca-Cola. What is it used for? It's used to clean. You see, if they have some injured people, as they get inside the car, blood will be spilled out on the seats. And so they use that as a good cleansing agent. You can even use that to clean your toilet bowls. So that is just to show you how strong is the phosphoric acid there. So it is generally carried by the name of soft drinks. So there's nothing soft about it. It's harder than the hard drinks that you know. And it is deadly. Because you become addicted to it. You just keep on using it and using it. And obesity sits in. After obesity sits in, cardiovascular disorders comes in. Okay, we'll talking about that when we go into that part. So let's continue. So your body oscillates from being fed, or starved. Now, we have programmed ourselves to eat 3 full meals a day, morning, noon, and evening. And there are people who only take food twice, the noon meal and the evening meal. They don't take their breakfast. However, it's been written about breakfast. And they said that the most important meal is breakfast because you have fasted almost 12 hours. When you go to sleep, and in the morning, you need to replenish the fuel of the body. But there are those who only take coffee and they are gone. They're all good after taking coffee. And then they have their meals, the noon and the evening meal. Now, you might be wondering why I always say noon and evening meal or morning meal. I don't say breakfast, dinner, and supper. Why? Because there's already a big change. During our time, our noon meal is known as dinner. And after dinner, which is the noon meal, is known as supper. Now it has been changed. The noon meal becomes lunch, and the evening meal becomes dinner. So those who are Christians, Christ did not have what you call the last dinner. It's the last supper, di ba? So why is supper removed and change it with dinner? When dinner is supposed to be a noon meal. Because we copied the Americans. Di ba? And we tend to be more Americans than the Americans. So whenever they say lunch, they mean dinner. And when they say dinner, they mean supper. All right, let's go back. So there's an alternate feed state and starvation state, or not really starvation, but fasting state. Fasting state. So after your meal, let's say breakfast, and your next meal is the noon meal, which is dinner. In between is fasting. Huh? And then you take your noon meal, and the last is supper. In between is lunch. Diba? Alright. Okay. Now, in the feed state, there's plenty of glucose. And that glucose is used by most of the cells of the body. And certain cells or certain tissues can utilize all of the three major metabolic fuels. I'm referring to carbohydrate, lipids, and proteins. Okay? But there are cells in the body, like your red blood cells, that cannot use any fatty acids or lipids or protein for that matter. It only uses glucose as its fuel molecule. And the way it uses glucose is very extravagant because the glycolytic pathway that it is using is anaerobic. Red blood cells does not have aerobic glycolysis. It is very paradoxical when we say anaerobic, it does not use oxygen, when in fact RBC are carriers of oxygen. And yet, they cannot use the oxygen. The reason for that is that it does not have a mitochondrion. It does not have a mitochondrion where your respiratory chain is located. Why is the RBC that does not have a mitochondrion? When the RBC was born, when it was created, we call them immature RBC. They have all the organelles. They have mitochondrion, Golgi apparatus, lysosomes, ribosomes, they have everything. But as they continue to mature, all of these organelles inside the cell are removed. Why? Why are they removed? In order to give space for a very important molecule, and that is the hemoglobin. Your red blood cell does not have only one hemoglobin. They have an average of about 200 to 240 hemoglobin molecules per RBC. And so there is no room for the organelles. So it has been removed. So the way that they are utilizing glucose is by glycolysis. And glycolysis is found in the cytoplasm. And so what is not removed are the enzymes for glycolysis. But because there is no mitochondrion, the glycolysis is dependent on anaerobic glycolysis without the use of oxygen. And it is very expensive in the sense that per molecule of glucose, it will only form 2 molecules of ATP. Very costly. Whereas if there is it is the anaerobic glycolysis which goes into the citric acid cycle, and then to the respiratory chain, you'll have plenty of ATP that will be formed out of one molecule of glucose. Now, cancer cells are like that also. That is why cancer cells likes to have so much carbohydrate, so much sugar, the better. Okay? So we will talk about that cancer cells in the latter part of our study of biochemistry. All right. There are cells like Dr. Ryan's lecture, the heart muscles or the cardiac cells can use ketone bodies, it can use fatty acids, and it can use also glucose. Now the idea there is that there should always be supply of ATP, whether it is the fuel is ketones, or the fuel is fatty acid, or the fuel is glucose. So there's always a constant supply, because we cannot afford to have the heart stop its contraction, because that contraction of the heart requires energy. So that is where ATP comes in. That's why the heart muscles can use any kind of fuel. All right. Formation. Okay, we will be talking about insulin, glucagon, and diabetes mellitus. Well, this is the glucose is controlled. The amount of glucose in the blood is controlled by insulin and glucagon. Insulin is secreted whenever there is a high concentration of glucose in the blood. And glucagon is secreted when there is a low concentration of glucose in the blood. And if you look at where these two hormones are produced, they are produced both in the pancreas, of course, but two types of cells, alpha cells and beta cells. All right. So the highlights of Langerhans, you have your alpha cells and your beta cells in your pancreas. And both of these hormones respond to the fluctuations of the sugar content of the blood. When the sugar content of the blood is high, then you have insulin that is secreted. If the low concentration of the blood is present, then glucagon is secreted, so that the glucagon will, of course, stimulates the formation of glucose from non-glucose sources, which is done in the liver. so that you will have a process known, or a pathway which is known as gluconeogenesis, or the synthesis of glucose from non-carbohydrate sources. So what are these non-carbohydrate sources? Foremost are the glucogenic amino acids that can become glucose. And then you have your glycerol. So where is this glycerol? This is when neutral fat or your triacyl glycerol is broken down and releases the glycerol as well as the fatty acids. And this glycerol can become your, or can be utilized to form glucose through gluconeogenesis. Okay? Pathways that process the major products of digestion. Okay. We have this. So, all right. So we have here carbohydrates, protein, and fat constitutes our diet. Okay? And they are digested by its digestive enzymes for carbohydrate and for protein and for fats. And the products for carbohydrate is simple sugars, mainly glucose. You say mainly glucose because isomers of glucose like mannose and galactose and fructose are converted to glucose. And then of course, digestion of protein is amino acids. And digestion of fat forms fatty acids and glycerol. So this is the absorbable molecules after digestion. All right. Then it is catabolized. Glucose, amino acids, fatty acids are metabolized in different pathways, and they have end products similar. Acetyl-CoA, in which the acetyl-CoA is transported into the mitochondrion. Why? Because in the mitochondrion, there is your citric acid cycle. Oh, citric acid cycle. So acetyl-CoA from fatty acids, acetyl-CoA from amino acids, simple sugars from carbohydrates into acetyl-CoA, they are all taken up by the citric acid cycle to produce reducing equivalents, Now, reducing equivalents in the form of NADH plus H and FADH2. Okay? And they are all taken up by the respiratory chain or electron transport chain to form ATP and water. Now, carbon dioxide is released here in your citric acid cycle. Okay, so all happens inside in the mitochondrion. The citric acid cycle is in the matrix of the mitochondrion. This respiratory chain is in the inner mitochondrial membrane. Now for conversion of fatty acids to acetyl-CoA also happens inside the matrix. The enzymes for that are collectively known as the beta oxidation enzymes and convert fatty acids into acetyl-CoA. And acetyl-CoA is fed into your citric acid cycle. Okay? So, you have different pathways for carbohydrate to become acetyl-CoA, amino acids to become acetyl-CoA, and fatty acids to become acetyl-CoA. They have different pathways. And we'll be studying those pathways. Okay? Carbohydrate metabolism is centered on the provision of feed of glucose. Glucose is the major fuel of most tissues. Metabolized to pyruvate through glycolysis, anaerobic tissues convert it into acetyl-CoA. Going back to RBC, it is anaerobic and the product is not acetyl-CoA. The product is lactic acid. Okay, so it's only aerobic glycolysis converts it into acetyl-CoA and enters the mitochondrion to be processed in the citric acid cycle to form carbon dioxide, water, and the reducing equivalents NADH and FADH2 goes to the electron transport chain to form ATP. Process known as oxidative phosphorylation or formation of ATP to oxidation. There is, however, a formation of ATP which is not oxidative. We call it substrate level phosphorylation. And this is found in the citric acid cycle itself. It forms for every turn of the citric acid cycles. Since it is a cycle, for every turn, you form one ATP, substrate level phosphorylation. Okay. All right, so here we are. This is an overview of okay, so from the diet, from the food that you eat, and it is digested to glucose and absorbed, and then you go into glycolytic pathway. This is glycolytic pathway. So this is aerobic because this is acetyl-CoA. Now, if it is not aerobic, then this goes to the formation of lactic acid here, lactate. Okay. All right. Now, when there's plenty of glucose, all of that goes, of course, if it is aerobic, into the citric acid cycle. But when there is plenty of glucose, the citric acid cycle becomes saturated. It cannot receive any more acetyl-CoAs because it's so full. So what happens if there's plenty of glucose because you eat too much? Extra rice. Huh? So this glucose is still oxidized, but there is no more ATP formation. It does not go down to acetyl-CoA. That goes into another, we call it hexose monophosphate shunt or pentose phosphate pathway. It is a shunt. See? Pentose phosphate pathway, or also called hexose monophosphate shunt, where you form two important molecules. This is NADPH, which is a reductant. Still remember the reductant used for cytochrome P450 comes from here, no? Pentose phosphate pathway. And this reductant, NADPH, is not only used by cytochrome P450, but it is also used for anabolic pathways. So what we mean by anabolic again? From smaller molecules to bigger molecules. So acetyl-CoA can be formed into fatty acids, long chain fatty acids, because acetyl-CoA has only 2 carbons. And if you require to have a fatty acid with 14 or 12 carbon atoms, so you need to have several of those acetyl-CoAs put together. And it is the NADPH which is utilized so that they can be put together to become a long chain fatty acid out of 2 carbons acetyl-CoA. And this is also similar in the formation of cholesterol. Cholesterol comes from acetyl-CoA. So when there's plenty of glucose and this is now saturated, that acetyl-CoA is used to make into cholesterol. Okay? Cholesterol, acetyl-CoA to become triacylglycerol, neutral fat. Now, so that is NADPH. Another important product of this pentose phosphate pathway is ribose phosphate. Ribose phosphate is used for forming nucleotides. And nucleotides are formed into RNA and DNA. Okay? So what is the implication of this? A cell cannot subdivide unless you form another DNA. Diba? You have this in your kindergarten chemistry, diba? No? You form DNA. So when there are 2 DNAs already in the cell, the cell subdivides into 2, each one having its own DNA, which is an exact copy of the original DNA. And you cannot have that unless you have nucleotides. And nucleotides cannot be formed unless you have ribose phosphate. And where do we get it? Through the pentose phosphate pathway, and that comes from glucose phosphates. You see? So when there's plenty of glucose, cells are subdividing, growing, multiplying. Now, that's why you have growth. But now you cannot grow taller anymore. You have attained your height. And if there's plenty of glucose, you still grow, but it is no longer vertical. It becomes horizontal. Huh? So you form a very bad word, bill. Make it a nice love handle. You call it love handle. Okay, so this is for growth, DNA. And the other one is NADPH, which is used in order that small molecules can become larger molecules when put together, courtesy of NADPH. So when this pentose phosphate pathway is operational, meaning you have plenty of glucose that you cannot use anymore. So you deposit it somewhere else by forming neutral fat, by forming cholesterol. You see? So when you have hypercholesterolemia, please advise your patient that they should minimize their carbohydrate. Minimize their carbohydrate. And do not blame the fat. That's what they said. It's the fat. It's the saturated fat now, which is wrong. Because when you have plenty of fat, when your diet is plenty of fat, cholesterol formation does not occur. It inhibits. If your food is plenty of cholesterol and fat, it inhibits cholesterol formation. If you have so much fat in your diet, it inhibits the formation of this triacylglycerol. But your glucose is not inhibited. It will always form triacylglycerol and cholesterol. Okay? We will go into that when we take up these pathways for this. No? Very interesting pathway. So don't forget it when you are being practicing and when you advise your patient because they have high cholesterol. Please do not say, do not eat fat. Just say, limit or do not eat carbohydrates anymore. Okay? All right. Lipid metabolism is concerned mainly with fatty acids and cholesterol. So I have said earlier that fatty acids are oxidized to acetyl-CoA through beta oxidation, which is located in the matrix of the mitochondrion, and that acetyl-CoA is fed into the adjacent citric acid cycle to form ATP and reducing equivalent NADH and FADH2, which goes directly to the inner mitochondrial membrane so that it will be oxidized to form ATP and water. No? All right. Okay, this figure here on the right side, okay, tells us what happens to the acetyl-CoA. And acetyl-CoA has three phases where it will become, will be oxidized to carbon dioxide. Acetyl-CoA can be formed into cholesterol, and in the liver, because of plenty of acetyl-CoA, can form ketone bodies. No? So here it is. From the diet, fats, fatty acids. It is esterified with cholesterol, glycerol, esterified with glycerol, and becomes triacylglycerol. And this is the one that is deposited in your adipose tissue cell. Now, by the way, cell growth cells do not grow or could not multiply more when they find themselves already tightly packed. So they do not grow anymore. But your adipose tissue cell is different. Your adipose tissue cell is different. They are not regulated by pressure of the next cell. They grow as fat as they want to be. No? So that's it. You can put in so many fat or neutral fat or triacylglycerol into your adipose tissue cell. Any amount, they will receive it. And of course, you suffer the consequence of growing sideways. All right. When there is a time when there is no more food or there is no more supply, then this triacylglycerol is being broken down to fatty acids again and glycerol. And that fatty acid is used in the mitochondrion. No? Fatty acids becomes acetyl-CoA through beta oxidation, and the acetyl-CoA is fed into the citric acid cycle. Okay. So acetyl-CoA, could you have source from carbohydrates and amino acid, becomes acetyl-CoA, and to be fed into the citric acid cycle. See this? Or acetyl-CoA, when there's plenty, as we said, when there's so much coming from here and in there, so this is citric acid cycle becomes saturated. So this acetyl-CoA goes into the formation of cholesterol. Okay? Or because there's plenty of fatty acids broken down, beta oxidation, more of acetyl-CoA, ketone body formation, ketogenesis. Huh? Now, of course, don't be afraid of your cholesterol. Cholesterol is a precursor of very important molecules, which is your steroids. So what are steroids? Your sex hormones, no? Male hormone and female hormone, without which we cannot define you, who you are, whether you're male or female. So that's the way. Okay? Now, this cholesterol is just a bystander. This is a very important molecule. No? Now, by the way, your liver is constantly producing cholesterol, whether you like it or not. 2 grams daily. How many milligrams is 2 grams? 2,000 grams.
2000.
Yes, 2,000 grams. And here you are, you're afraid when your cholesterol goes to 180 or 200 milligrams. 2,000 milligrams, I'm sorry, not 2,000 grams. 2 grams is 2,000 milligrams. And of course, when your cholesterol level in the blood is about 120, 200, and you are very, very afraid already when it reaches 250, and yet your liver is producing 2 grams, and that's 2,000 milligrams. It's because it is converted into other products. If you still remember your cell membrane has cholesterol, without which that cell membrane does not function. How many cells do you destroy every day? How many are being replaced? And that cell membrane requires cholesterol. That's why you produce that much, 2,000 milligrams or 2 grams daily. So this cholesterol is just more or less, this is not the criminal na pagbintangan lang, no? Napagbintangan lang because it is carried by a bad carrier, LDL. If this cholesterol goes into the HDL, then it becomes good. But it just so happened that it went into a bad company, LDL. So it becomes bad. Huh? So do not call it bad cholesterol or good cholesterol. Cholesterol is neither good nor bad. It is just that when cholesterol is carried by LDL, no, and you know LDL is found in the atherosclerotic plaque. You do not find HDL there. It's the LDL. And naturally, if you have cholesterol, so it is cholesterol. So they put the blame on cholesterol. They do not put the blame on LDL. Cholesterol is just only carried by the LDL. Nothing to do with the problem of atherosclerosis. It just happened to be in a bad company, and that's the LDL. Okay? All right. So again, we have this. It can be oxidized. There you are. Carbon dioxide and water by the citric acid cycle forming juicing equivalent, which goes into this respiratory chain or the electron transport chain to form ATP and water. No? It is a precursor of steroids. It is also the precursor of ketone bodies, acetoacetate and hydroxybutyrate. Okay? Much of the amino acid metabolism involves transamination. So removal of the amino group performed by transaminases, group 2 enzymes or class 2 enzymes, transferases. Okay? So this we were talking about enzymes yesterday. You have your alanine transaminase, no? ALT. So you have your transaminase there. It moves an amino group from alanine. Okay. So the carbon skeleton, that is your amino acid minus the amino group, could be either utilized for oxidation or utilizing it in your Krebs cycle or the citric acid cycle, or it can also be used for forming back the amino acids. As I have given you example of glutamic acid. So glutamic acid minus the amino group is alpha ketoglutarate, and intermediate in your citric acid cycle. So amino acids required for protein synthesis. That's the most important function. So there are two kinds, sure, essential and non-essential. So it does not mean to say that the non-essential are not needed. All of the 20 amino acids that we have learned are needed, very much needed. They're only called non-essential because the body can form it. So it does, it means that it need not be present in your diet. But the essential, we cannot make this. We cannot make this. So if we cannot make this, then it should be in our diet in order to have it. Now, why did God or your creator have this essential or non-essential? No? To make you perfect, the non-essential na lang tanan. Now, it's like this. The essential amino acids, the formation of these essential amino acids require several enzymes. So several steps before you can form. But the non-essential, some it is just a one-step reaction. You form the non-essential already. Or one step or two step, at most three steps. But some of these essential require 17 enzymes, no, in order to form it. So 17 reactions. So it is much easier to get it from our food because we need food, fuel for our body. We are not self-contained. We are not like plants, but even then plants get its nutrient from the soil and water and the energy from the sun. No? And that energy and that removal of or the procurement of nutrients from the soil and the water is all the time. But us, we only eat when we are hungry. And some of us even eat if we are not hungry. No? Look at the animals. They only eat when they are hungry. But the plants, they have to get their nutrients all the time from soil. And that's why they cannot get out from the soil. Otherwise, they cannot get any more. If they get out from the soil, they could not have nutrients anymore. They die. So plants are fixed. Okay? Now, what do we do with the removal of the amino groups from our amino acid? We have a pathway which forms the amino group into urea. Why? Because the amino group that is removed from the amino acids becomes ammonia. And ammonia is toxic to the brain, to the nerves, and you'll have a condition known as hyperammonemia. So in order to remove that toxicity, that ammonia is converted to urea. If you pickle your liver by alcohol and cannot perform this, then you will have hyperammonemia. That usually happens in cirrhosis of the liver. No more urea formation. You will have hyperammonemia. And of course, death follows. Diba? All right. Okay, so removal of the urea, or removal of ammonia into urea, and the carbon skeletons are converted or oxidized to carbon dioxide through your citric acid cycle, okay? Or Carbon skeleton will be utilized to synthesize glucose through a process known as gluconeogenesis, or form ketone bodies, or acetyl-CoA, which may be oxidized through the citric acid cycle again, or used for synthesis of fatty acids. That's acetyl-CoA. Now, so the carbon skeletons of amino acids, carbon skeletons or amino acids minus the amino group could either be directly oxidized in the citric acid cycle, can be used for forming glucose, can be formed into ketone bodies, or the acetyl-CoA can be formed into fatty acids. Okay? Here you are, you have your protein diet. All right? Digestion, amino acids, they are absorbed and they can become tissue proteins and also plasma proteins, other important proteins, and they can be degraded and they can become amino acids again. Or these amino acids can become non-protein nitrogen derivatives. So example like your formation of your thyroid hormones from your amino acid tyrosine. No? Okay. All right. You have carbohydrate can be formed into carbohydrate. Now, the carbon skeleton can be formed into glucose, or it can be become again amino acid. Okay. Or amino acids like the ketogenic amino acids becomes ketones, right? So after transamination of this amino acid, becomes acetyl-CoA, goes into the citric acid cycle, carbon dioxide is formed. Or after that removal of the amino group, the amino nitrogen in glutamic acid, okay, deamination and forming urea, or elimination of the amino group. So metabolic pathways may be studied at different levels: tissue and organ level and subcellular level. Two areas: organ level or subcellular level, like the organelles. For example, your mitochondrion, where you have your beta oxidation, where you have your citric acid cycle, where you have your electron transport chain. Or in the compartment known as the cytoplasm or cytoplasmic compartment, where you have the glycolytic pathway or your glycolysis. Okay? At tissue and organ level, okay, resulting from the digestion of proteins, glucose from carbohydrates. So on the liver has the role of regulating blood concentration of these water-soluble metabolites. And we know that this glucose, you have your insulin and your glucagon, which is responsible for maintaining a glucose concentration in the blood. And then when there's plenty of glucose, okay, they are first stored as glycogen, where stored as glycogen in the liver and in the muscle. Or, look at this, fatty acids. So that is your glucose in excess of immediate requirements are stored as glycogen or fatty acids. And in between meals, the liver acts to maintain blood glucose by breaking down glycogen. And if this fasting continues, then the fats are broken down. Lipolysis. Okay. And of course, gluconeogenesis, formation of glucose from non-carbohydrate sources, especially from amino acids, glucogenic amino acids, and glycerol. Okay? So this is where you have your all right. So where do we start? We start from the intestine. Okay? The intestine. This is transport of lipids. All right, in the intestine, since triacylglycerol is not soluble in water, it is packaged by what we call lipoproteins. And the lipoprotein coming from your intestine is the chylomicrons. Okay, so it carries that. TG is triacylglycerol, and you have your lipoprotein. Okay. Now, by the way, your liver is also producing fatty acids out of the conversion of glucose into fats. See, glucose to fatty acids, esterify it with glycerol, you'll have triacylglycerol, and give it to the blood. And since the blood is aqueous solution, it has to have a carrier, and the carrier is VLDL coming from the liver. And we have your VLDL. It carries the triacylglycerol as well as the cholesterol that is formed by the liver. So here you are, here now. So this can be given to the muscles and to the adipose tissue. See? There. So the fatty acids here can be utilized or oxidized, and you have your carbon dioxide that will be formed. Okay, the fatty acids, lipoproteins, again glucose becomes fatty acids, and fatty acids can go into the liver here and to the muscles here. NIFA is non-esterified fatty acids. Non-esterified. So now your cholesterol is esterified at its number 3 carbon atom, which has an OH. The OH there is the site of esterification. So it is esterified. Because cholesterol cholesterol is considered as an alcohol. That's why it's cholesterol, all. Remember that in your organic? Anything that ends in all is an alcohol. Okay. And how do you form esters? You form esters with an alcohol and an acid. Okay? So non-esterified fatty acid. It is not esterified. No, so we have alcohol, cholesterol. You add a fatty acid to the OH, the number 3 carbon of cholesterol, then you have now an esterified cholesterol. All right. So it's just muscles can use it. The fats, when glucose is not it's not available right away, then you can use the fat in the muscles, oxidize it. Adipose tissue is main fuel reserve of the body. Remember this. It is the main fuel reserve of the body because the glycogen stored in the liver and the muscle are the first one to be used. That's why you need your 3 meals every day to replenish that glucose reserve. And if you do not replenish it, then fats are utilized. So one of the ways of removing your fat is fasting. But fasting is, although if you are Christians, you are being told to fast and pray. Fasting is very, very important among Christians. No? But sometimes we forget that we are Christians. We don't fast, we feast. Instead of fasting, we feast.
Tama.
Prayers. It does not go together. Fasting and prayer goes together, but not feasting and praying. Okay. Now we will have plenty of this VLDL, this chylomicrons, this LDL, this HDL, when we take up this metabolism of neutral fat, special fatty acids, no? At subcellular level, glycolysis occurs in the cytosol and citric acid cycle in the mitochondria. So glycolysis, whether aerobic or anaerobic, is cytoplasmic, while the citric acid cycle is mitochondrial. Now, before it can go into the mitochondria, it has to be acetyl-CoA. This is just to show the importance of, this is your mitochondrion, no? That's the outer membrane, the inner membrane. And these are your projections inside, no? Thrown into folds, the inner mitochondrial membrane. Okay. So, from glycolysis, when you form pyruvate, pyruvate enters the mitochondrion. Pyruvate enters the mitochondrion. Why? Because the enzyme that converts pyruvate to acetyl-CoA is, and it's acetyl-CoA there, is found inside the mitochondrion. And that's your pyruvate dehydrogenase. So it's here. So it has that pyruvate from, this is glycolysis, and you have pyruvate, then goes inside the mitochondrion, and acetyl-CoA becomes citrate, together with oxaloacetate becomes citrate, citrate becomes alpha-ketoglutarate, alpha-ketoglutarate becomes succinyl-CoA, succinyl-CoA becomes fumarate, fumarate becomes malate, malate becomes oxaloacetate again. Huh? Okay, so this is just to show you the importance of your mitochondrion. Now, aside from the citric acid cycle, we have inside the matrix also for the metabolism of fatty acids, and that is the beta oxidation. And the product is still acetyl-CoA. So acetyl-CoA goes into the citric acid cycle. It's very easy because the fatty acids are oxidized in the enzymes present in the matrix known as the beta oxidation enzymes. Okay, so there is regulatory. The flux or the flow of metabolites through metabolic pathways must be regulated in concerted manner. We have here what we call allosteric enzymes. Allosteric enzymes are regulated through its allosteric site. What do you mean by allosteric site? It's the other site other than the catalytic site of the enzyme. So that other site could be a regulatory site of the enzyme. So you have regulatory enzymes. Okay, non-equilibrium reactions are potential control. Non-equilibrium. This is equilibrium. No? Equilibrium. Called the steady state condition. So net flux from left to right. No? Uy. Naglubat ako. Now, this here, you can say that there is a flow, no? Because A to C is also can be C to A, and C to D can also be D to C. Unless you are going to remove this, remove that, then that can go forward. Because the C becomes D, and this becomes smaller in concentration. So A will be converted to C. But in this state, when D is not removed, then it's just only like that. There is no flow. But once you remove this, then the direction will be going towards this. If you remove this, then the direction is going leftward. The flux generating reaction is the first reaction in the pathway that is saturated with the substrate. Okay, let's try to explain this. All right, what he's trying to say is this one. This is the cell membrane. Okay. Now, number one is to allow A to pass through. No? Okay. Once the A is here, then this is reversible, A to B, B to A. And that B can go to C, and that C becomes D. But there's plenty of this, no? Then it is going to stop this enzyme here. You know that, no? And this could also stop this enzyme here. Now, this is what we call a feed forward. The presence of A increases the enzyme here, and that's activity. This is a feed forward, allosteric. This one is negative feedback on this particular enzyme. All right. This one is for activating this enzyme. It's either calcium calmodulin or cyclic AMP that activates this enzyme. Okay? All right. This regulatory mechanism, this 1 and 2 are fast in regulating right away. But there are also slow regulatory mechanisms. And this is in the synthesis of the enzyme. Okay, so ribosomal synthesis of enzyme. There. Excuse me. So the enzyme has to be synthesized first. There you are. So although this can also regulate, but it is slow, takes time in order to have that enzyme. Okay. Oh, it's somewhere here. Oh, here we are. 1 and 2. So this is 1 and 2 are rapid mechanism of regulation, whereas 3, 4, and 5 are slow because you have to synthesize the enzyme first. You have induction, and then you have repression of the nuclear formation of messenger RNA. And the messenger RNA is read or translated by the ribosomal RNA or the ribosomes before you can make the enzyme. So this is a slow regulation, while this one is very fast, either by phosphorylation or dephosphorylation of the enzyme. Where is that? Do we have it here? Oh, here. Involving phosphorylation and dephosphorylation reactions. Now we will be able to see this when we take up glycogenesis and glycogenolysis. It's a very good example of this phosphorylation and dephosphorylation reactions. Many metabolic fuels are interconvertible. Okay. So meaning it can be interconverted, like fatty acids into ketone bodies, no? And fatty acids into acetyl-CoAs. And acetyl-CoAs to become cholesterol or fatty acids again, interconvertible. Now, however, fatty acids, because the only way that the fatty acids are oxidized is through your beta oxidation, and the product is acetyl-CoA, you cannot use that for making glucose. However, there is portion of the fat that becomes a source for making glucose. Remember that you have your triacyl glycerol, meaning you have a glycerol to which three fatty acids are attached. So that's why it's called triacyl glycerol. Now, once that is lipolyzed, fatty acids are released, and glycerol can be used for forming glucose. You see? That's the only part of the fatty acids. But the rest of the fatty acids cannot be converted to glucose. Now, and another thing, the acetyl-CoA that is formed from pyruvate is irreversible, cannot be reverted back to pyruvate, in which pyruvate can become glucose. No? But because this reaction of pyruvate dehydrogenase to form acetyl-CoA is irreversible, you cannot convert that to glucose. So the only part of your triacyl glycerol that can become glucose is your glycerol portion. So here we are again, feed and fasting states. Now, erythrocytes lack mitochondria. So they are dependent on anaerobic glycolysis. The brain can metabolize ketone bodies. Okay, now, there's a good thing here. Whenever glucose is low, now remember that your nervous tissue, brain, and the nerves prefers to use glucose. However, when glucose is low, they revert to the use of ketone bodies. Why? Because when you are fasting or in a fasting state, the cells now utilizes fatty acids as fuel. And as a result, you will have plenty of acetyl-CoA. And acetyl-CoAs at the fasting state cannot be made into cholesterol. Remember that. Why? Because there is no glucose. So what is glucose there? Remember when I said when you have plenty of glucose, a pathway is activated known as pentose phosphate pathway. And the product of pentose phosphate pathway is NADPH, and formation of cholesterol from acetyl-CoA utilizes NADPH. See? Even if you have plenty of acetyl-CoAs, because there will be plenty of acetyl-CoAs during fasting state. Breakdown of fatty acids to acetyl-CoA will be plentiful during fasting state. But that acetyl-CoA cannot be made into cholesterol because there is no NADPH, and you can only form NADPH when there's plenty of glucose. Now in this case, now the state is fasting, so there's no glucose. Okay, so in that case, in which the brain likes to use glucose, there is no more glucose, so it reverts to ketone bodies. Utilization of ketone bodies. But the red blood cell cannot go on without glucose. That's the reason why other tissues goes to the utilization of ketone bodies and fatty acids in order to preserve whatever glucose is left, and that will be utilized only by the RBC. Although there is gluconeogenesis going on, or the formation of glucose, it is not enough. But it is enough for utilization of by the red blood cells. Okay? So the reason why other tissues revert to the utilization of other fuel other than glucose is to preserve the glucose for RBC, to reserve it for RBC. Metabolic interrelationships. Here we have adipose tissue, the blood, and the liver. You can just easily see here, you just follow the lines, but you know, it is hard to do this because you have not studied the different pathways. But once you have studied the different pathways, this particular illustration is very, very helpful. Now, I do not want to talk about this yet because this will confuse you. No? And we are confused, we need Confucius. Okay? So let's preserve this after we take up the metabolic pathways. First, carbohydrate and lipids, and then amino acids. And then we go back to put them all together. In the phase state, metabolic fuel reserves are laid down. Now, if you look at this, now this table, as you can see, energy yield, oxygen consumption, carbon dioxide produced, respiratory quotient, and energy here. Now, look at this. Carbohydrate. Okay. Let's go right away to RQ, respiratory quotient. Amount of oxygen consumed, no? And carbon dioxide produced. So, as you notice, it's here, no? 1. Lowest is about 0.8 and 1, no? I'll just put it as 1. Meaning to say, 1 carbon dioxide, 1 oxygen. Now look at fat. To use it, look at this. 0.7. So you consume more oxygen to produce carbon dioxide, 0.7. And very near to alcohol. Okay? 0.6 carbon dioxide, and you use 1 mole of oxygen. And you only have 0.6 of carbon dioxide. So this is costly alcohol. You need more oxygen in order to metabolize it. You look at your respiratory quotient. So it is easy to metabolize carbon dioxide. A little bit harder for protein and still harder for fat and very hard for alcohol. So this is what is meant by this. You have your respiratory quotient. Okay? Oxygen consumed 0.8. Carbon dioxide produced 0.8. So similar. And quotient RQ is 1. So 0.8 over 0.8 is 1. No? So that is, here we have 0.9, and you have only, you use almost 1 molecule of oxygen, and you have only 0.7 carbon dioxide. There. Okay? So what is important is your RQ. Plasma concentration of metabolic fuels in the feed and fasting state. Concentration of plasma, metabolic fuels. No? Okay. Feed state 5.5. Fasting 3.6. 7 days starvation 3.5. So it is maintained. Fasting here for 7 days. So this utilization of, or this amount of glucose in the blood or in the plasma is maintained for 7 days. This is just only for the red blood cell use. All other tissues utilize already other so look at this. 40 hours fasting, your fatty acids have increased. No? 1.5, 1.1. From the feed state, it's only 0.3. Okay? 7 days, 1.1. So you are utilizing fatty acids. Ketone bodies, during the feed state, it is negligible. But 40 hours fasting, 2.9. 7 days fasting, 4.5. It's already increased. Okay. All right, summary of major metabolic features in principal organs. Okay, we have the liver. Major pathways in the liver, we have glycolysis, gluconeogenesis, lipogenesis, beta oxidation, citric acid cycle, ketogenesis, formation of lipoproteins, drug metabolism, cytochrome P450 here, synthesis of bile salts, formation of urea, again, this is very important, removal of ammonia, then uric acid from your xanthines, then cholesterol formation in the liver, 2 grams daily, then plasma proteins. So what are the main substrate of the liver? Non-esterified fatty acids, glucose in feed state, lactate, glycerol, fructose, amino acids, alcohol. Major products exported: glucose from gluconeogenesis, triacylglycerol VLDL transport, ketone bodies, urea, uric acid, bile salts, cholesterol, plasma proteins. Enzymes present: glucokinase, glucose-6-phosphatase, glycerol kinase, phosphoenol pyruvate, carboxykinase, fructokinase, arginase, HMG-CoA synthase, HMG-CoA lyase, alcohol dehydrogenase. The brain. So look at this. The brain has glycolysis, citric acid cycle, amino acid metabolism, neurotransmitter synthesis. What is its substrate? Glucose, amino acids, ketone bodies, prolonged starvation. Ketone bodies in prolonged starvation. Major products: lactic acid, as in products of neurotransmitter metabolism. Special enzymes, those for the synthesis and catabolism of neurotransmitters. That's liver and brain. The heart. Look at that. Pathways of the heart: beta oxidation. That's why it can utilize fatty acids, and citric acid cycle. We can also utilize glucose. Substrate: ketone bodies, non-esterified fatty acids, lactate, chylomicrons, VLDL, triacylglycerol, and glucose. No products exported because it is always using fuel. Special enzymes, lipoprotein lipase, very active electron transport chain. Naturally, because you'll have to have more ATP in order that that heart can continuously do its function. Adipose tissue, lipogenesis, esterification of fatty acids, lipolysis, and in fasting, so lipolysis occurs in fasting. So adipose tissue degrade its triacylglycerol in fasting. So substrates: glucose, chylomicrons, and VLDL triacylglycerols. Product exported: non-esterified fatty acids and glycerol. Special enzymes: lipoprotein lipase, hormone-sensitive lipase of the pentose phosphate pathway. See, important ang pentose phosphate pathway because it forms NADPH, and that is the one that is used for forming triacylglycerol to be deposited in adipose tissue. Fast twitch muscle, glycolysis. That's the fast twitch. So if you develop your fast twitch muscle, it will always be glycolysis. Fast twitch is very fast, but only a short period. And compared to continuous muscle contraction like in marathon running, no? So you have the slow twitch muscles for marathon runners. What do you have? Citric acid cycle and beta oxidation. Yeah, burning fat. So substrates, ketone bodies, chylomicrons, VLDL, triacylglycerol. No exported products. Very important enzymes, lipoprotein lipase, very active electron transport chain, because that is a long contraction. Marathon runners or marathon swimmers. Kidney is gluconeogenesis. So this is one organ that can form glucose. Oh, yeah, gluconeogenesis. Aside from the liver, it's the kidney. So substrates, glycerol, glucose, lactic acid, and non-esterified fatty acid exports glucose because of gluconeogenesis. And especially special enzymes, you have glycerol kinase, phosphoenol pyruvate carboxykinase. Then you have erythrocytes, anaerobic glycolysis. Look at that. And pentose phosphate pathway. Then you have glucose, then you have lactic acid as export, hemoglobin, enzymes of pentose phosphate pathway. So those are major organs and subcellular organelles. Okay. So what do we have? Essential tissue proteins. So death results when essential tissue proteins are catabolized and not replaced, as in cachexia. A result of release of cytokines in response to tumors and disease. There is an increase in the rate of tissue protein utilization, as well as considerably increased metabolic rate. So they are in a state of advanced starvation. Again, death results when essential tissue proteins are catabolized and not replaced. All right. For fetosis, there is a high demand of glucose. And lactose synthesis in lactation can lead to ketosis, a mild ketosis, with an increase of glucose in lactating mothers. Now, cattle AOS carrying twin pregnancy, very pronounced ketoacidosis, and very low glucose because it is being utilized very, very fast, like twins. So this is, as you see in cattle and iwis, this is rare to have twins. Pronounced ketoacidosis and hypoglycemia. Because the way they use cattle eat grass. And what is in grass? Cellulose. So before they can utilize that cellulose, they have to ferment it. Fermentation of cellulose in order that you will have short chain fatty acids like acetic acid, propionic acid, and butyric acid. And those short chain amino fatty acids are being utilized through beta oxidation. Your acetic acid can easily be turned into acetyl-CoA, and it goes to the citric acid cycle right away. And then there is a high demand for lactose because that is the food for the newly delivered animals, mammals. So that's why glucose would go down very, very fast. Hypoglycemia. Well, they have diabetes mellitus. We'll be talking about this. No? You know already the type 1 diabetes mellitus. That is, there is no insulin, but in the type 2, there is insulin, but there is less sensitivity of the tissues. When we say less sensitivity, the insulin there cannot get inside the cell because insulin being a protein, it has to pass through a lipid bilayer. A lipid layer, lipid bilayer. That's your membrane. And you know that your insulin is water soluble and the membrane does not allow that. So it needs to have receptors. So what is meant by tissue not sensitive? Because that tissue loses its receptors. So if you do not have receptors for insulin, that insulin cannot be taken into the cell. So that's the problem of your type 2 diabetes. There's plenty of insulin, but it cannot get inside the cell because of the problem of the receptors for insulin. Okay. All right. We will be talking about pathways after the lecture on your fuel molecules. And your fuel molecules are we have taken up already proteins and amino acids. So what is need to be lectured would be carbohydrate of physiologic significance and lipids of physiologic significance before we embark on your metabolic pathways. First with metabolism of carbohydrates. We will be talking about glycolytic pathway. We will be talking about hexose monophosphate shunt or pentose phosphate pathway. Then you have your glucuronic acid pathway. No? So on other pathways, so we will have glycogen formation, glycogenesis, breakdown of glycogen, glycogenolysis, and production of glucose from non-carbohydrate sources, and that is gluconeogenesis. And then we will have utilization of the pathways that utilizes fatty acids. And first we will have beta oxidation for how fatty acids are converted into acetyl-CoAs and the formation of cholesterol